Good, how are you?
But I thought you were just like a contractor.
It's live.
It's not gonna fall, is it?
Last row, guys.
Hello, everybody. Please settle down. Thank you. Thank you for coming in and finding your seats. I appreciate that. Welcome to our families. Yeah. All right. We are all very excited to be here. All students should have enough space to be seated with an empty seat in between you and the next student. Yes? Yes. All students should have enough space to be seated with an empty seat between you and the next student. If anybody feels that they're not comfortable in the seat where they are right now, now is a good time to change your seat. Raise your hand if you have any issues with the seat where you are right now. All right. We are going to be here for periods 1 and 2. All right, 2 periods in this seat. Thank you. Great. All right, so I'm Ms. Cummings. I'm the school librarian here, and I have the pleasure of teaching 816 research portion of class, along with Ms. Williams, who teaches the research portion, as well as the art portion of the 816 class. I also have the pleasure of knowing so many of these 6th graders from the library and from Ms. Sorienti's class. Yes, I see your pass. Don't lose your pass. Put it away for later. Thank you. All right. And then we have Mrs. Leonardos, who teaches the 7th grade IIM class. Excellent work you're doing. And that's why we're here today, to celebrate all the great work that they're doing in their 716 IIM class. And we have Ms. Sorienti, who teaches the 6th grade IIM classes. And we're not going to keep talking. We have to settle down. And it's extra exciting because this is the first year that we have 5 IIM classes in the 6th grade. So the program is growing, it's expanding, the kids are learning a lot, it's exciting. Chester, we're not going to keep talking. If you're not in a good seat for you, you should move. Okay? Thank you. Okay. All right. So I'm going to turn it over to Mrs. Leonardos, who's going to introduce what's going to happen today and why we're here. Thank you all.
Thank you for coming. Okay, so we're really excited to be here. Leonardo, like Ms. Cummings said, I am the 7th grade science teacher and the blind teacher of 716. We take a science focus in 7th grade. We focus on stem cells for one portion of our year, and the other portion of our year we focus on forensic science. So for this portion that we're going to be presenting to you guys, we're going to be talking about stem cells. I'm not going to tell you what it is. They are going to teach you. So the way it's going to go is there's 5 students. We have Michael, Olivia, Simone, Jaden, and Sam, and they will be presenting within 10 minutes. So in those 10 minutes, they might take anywhere from 5 to 8 minutes to present, and then the remaining portion will be for questions from the audience. At the end of the 10 minutes, we're going to move on to the next presenter, and they will again have 5 to 8 minutes to present and then answer any of your questions. At the very end, we have a panel of students. As you can see up there, we have Smith, Melody, Billy, and Paris, and they will be answering questions that you have already given to us. If there are any other questions at the very end, and we finish a little earlier, you can ask our panel any questions about IIM, about the class, and if you have any questions about the presentations, you can ask the correct student that is going to be presenting. If there are any questions, you can ask me right now before we start. No? Good. So Michael will be the first to present. He will be talking about stem cells and stem cell research, and then the rest of the students will go after him. And they have very specific research questions that they're going to be presenting. All right, we're going to start with Michael first.
Hello, my name is Michael Hernandez from class 716, and my focus question is, how are different genetic editing tools more effective than CRISPR? And my thesis statement is, different genetic editing tools can be more effective than CRISPR because they can incorporate a stronger repair mechanism with more precise DNA changes and utilize a gene knockdown method. Before I talk about CRISPR, I first need to explain stem cells. Stem cells are undifferentiated cells. Undifferentiated meaning they can become any cell in the body. There are multiple types of stem cells, like somatic, embryonic, and induced pluripotent. Somatic stem cells can become any stem cell, but they are limited to what they can become. They can only become specific types of tissues. And embryonic stem cells can become any type of stem cell in the body, but they are very limited and hard to find. And induced pluripotent is when scientists try to turn the somatic stem cells into embryonic stem cells. The embryonic stem cells are found in a blastocyst. It's usually the first week after fertilization in the egg. They are taken out and used for research. So, we have CRISPR now. You may be asking, how does CRISPR relate to me? Why do I care? CRISPR, in the near future, CRISPR will and can be used to edit your genes, to change your appearance, height, or anything. If you want to change your color of your hair, you can do that. Also, CRISPR can delete multiple diseases. And CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. CRISPR edits genes using multiple tools like Cas9 and guide RNA. Cas9 is an endonuclease. An endonuclease is what helps gene editing tools make cuts and changes in the DNA. And guide RNA essentially acts as a map for Cas9 and CRISPR to find where they want to make this change. This process usually takes about 2 weeks to complete on average. In this chart, it shows the gene editing market in the future and right now. So in 2022, CRISPR shares 43.7% of the revenue in the gene marketing editing, in the genetic editing market. CRISPR is in the dark blue in the bottom, and it is compared to Talent, ZFN, and other genetic editing tools. Right there is the dark blue. Scientists seem to be investing more of their time into CRISPR instead of giving these other genetic editing tools a chance, because CRISPR is cheap, fast, and reliable. But CRISPR has a few flaws. A human trial done with CRISPR on patients with lymphosoma had a 60% success rate. 60% of these patients were fine, 21% got sick after a few months, and the rest were sick from the start. And these changes are not close either. These changes can be thousands of base pairs away from the intended target. Base pairs, 2 base pairs make up a nucleotide. Nucleotides are the lines in the DNA. CRISPR is also limited by protospacer adjacent motif, or PAM for short. PAM is a sequence of DNA that has to be recognized before CRISPR can actually make a change. But PAM is not found everywhere within the genome. So CRISPR can only make changes within a select few places in the genome. Alternative would be TN7. TN7, like Cas9, is an endonuclease, but it is a transposon. Transposon is referred to as the jumping gene, meaning it could go anywhere within the genome without the use of guide RNA. But in this situation, guide RNA tells it where to go, and it goes by itself. Then it makes a change, but instead of leaving it to be repaired by the DNA's natural repair system, the homology-directed repair, it inserts itself into the genome after an edit is made, leaving less room for errors and ensuring that it is safer for people to use. Another good gene editing tool to use is TALENT. TALENT stands for transcriptor activator like effector nucleases. And instead of using Cas9 as its endonuclease, it uses FOC1. FOC1 combines with tail proteins in order to make a change. But the difference with TALENT is that it is very precise. TALENT has an 88% success rate compared to CRISPR's 60, so it is 28% more accurate than CRISPR. TALENT is also not limited by PAM. PAM does not have to be recognized before TALENT has to make a change. So it can make a change everywhere in the genome. And instead of using guide RNA, TALENT uses the DNA binding domains, or PBDs. But one downside with TALENT is that it is very tedious to work with. This is because tail repeats have to be on each side of the DNA before it actually makes a change. So this requires double the cloning work. This process usually takes about 1 to 2 weeks, and most of the time is used to clone these tail repeats. Lastly, we have RNAi. RNAi, unlike TALENT, can perform a genetic silence. It is very easy to work with. It only takes 1 to 4 days to fully complete a gene knockdown. And you may be asking, what exactly is a gene knockdown? RNAi targets the mRNA transcript. The mRNA transcript basically tells the DNA how to make protein. And once it basically cuts it out, the DNA can no longer make protein, making sure that it won't work anymore, or it silences it. Scientists can measure the effectiveness of this gene knockdown by using Western blot and qRT-PCR. Western blot measures the amount of protein in the sample, and qRT-PCR measures the amount of mRNA in the sample. Both have to be low in order for scientists to ensure that gene knockdown has worked. But RNAi is a bit less precise than CRISPR. But with all these choices just go to show that there are many different alternatives instead of CRISPR, and in the near future, with these other genetic editing tools, you can ensure that you will be safer in the future when you want to edit your genes or child's genes. Yeah, that's it.
Give it up for Michael. Thank you. Oh, I forgot about the timing. One moment.
We have 4 minutes left. If anyone wants to ask any questions, Michael has the floor for 4 more minutes. Any 6th graders have questions? Now's the time.
Yes. Yeah.
What do you figure is the best test? One that's 88%?
Yeah, and from all the genetic dimensions that I mentioned, I personally think talent is the best one. What do you mean? Like cost to make? Or cost to like edit your genes? Edit your genes? I don't know the exact price, but I do know it will be very expensive. This will probably only be limited to very rich people.
We have a question back here.
How long will it be? So like, this is like not common, but like crazy surgery.
How long do you think this will be until this is more commonly used, about as common as LASIK surgery, or something like that?
About like 5 to 10 years until like they still need to. They're doing human trials right now, making sure that it's safe for humans to use and it will not like give them other diseases. So about 5 to 10 years.
Any other questions? Raise your hand. Miss Leonardo?
It depends on.
Can you repeat the question? What sicknesses, so everyone can hear?
If the editing fails, what's the most common sickness a patient will get?
Most common, but the sickness they get depends on the base pair that is mutated. The base pairs each base pair has a very specific role, and if one is mutated, then it could give many different types of diseases.
Any other questions? He has 2 more minutes.
Stand up, come on. We have a question. I'll proceed.
If you were to alter the genetics with CRISPR, would you have to do it in the embryo, or could you do it with like an already developed human?
Did everyone hear? She asked if you edit the genetics of a person, would it have to be in an embryo or already developed human? Is that what she asked? Yeah.
Well, it's easier in the embryo, but it can be done with humans to like take out diseases.
Would the results be like, how long would the results take for like the gene to
How long will the results take to see?
For the genes to be altered.
For the genes to be altered.
Like I said, like 2 weeks for CRISPR, 1 to 2 weeks for talents, and 1 to 4 days for RNAi. And then
Okay.
for TN7 is 2 weeks also.
Are there any other questions? All right, one last question, because we have a few more seconds.
What is a genome?
It's all the DNA in a cell or human body.
Or in anything, right? The genome of like a plant or what have you, but humans specifically now. Any other questions? Great, so we have our next presenter. Do you want to present them for us?
I would like to present Olivia, my classmate.
Hi, my name is Olivia of class 716, and my focus question is, how do bone marrow stem cell transplants affect individuals suffering from HIV? Bone marrow stem cell transplants affect individuals suffering from HIV by creating healthy mutations, preventing HIV from spreading, and stopping certain strains of HIV from recurring. HIV is a blood-given and sexually transmitted disease with a high mortality rate. When it was first recognized in the 1980s, approximately 100% of the patients who were diagnosed ended up dying. Currently, patients suffering from HIV have to take antiretroviral drugs so that the disease does not spread further through their body. There's currently no novel treatment to fully cure the disease, and the chart above shows in the 1980s the death rate of HIV. And shows also how it was mostly prominent with men. The first documented case of HIV being cured was the Berlin patient, or Timothy Ray Brown. He was diagnosed with HIV and acute myeloid leukemia, and he underwent a bone marrow stem cell transplant to help in treating his leukemia. And afterwards, tests were done on him that showed that the HIV levels in his body significantly decreased and they were no longer spreading. The key genetic variation associated with his apparent cure for HIV is the CCR5 delta 32 mutation. CCR5 is the gene that HIV binds to and uses to quickly spread through the body. This delta 32 mutation prevents HIV from being able to bind onto the CCR5 co-receptors. Only approximately 9% of people have this mutation, and only 1% of people have it in both of their CCR5 genes. This serves as a critical focus in HIV research as it provides a window for an opportunity for a cure in HIV. Now, there are a lot of benefits with the stem cell transplant in helping curing HIV. One being that studies done by the National Institute of Allergy and Infectious Diseases showed that getting a bone marrow stem cell transplant can significantly reduce the HIV levels in someone's body, and also bone marrow stem cell transplants can create new immune cells that are HIV resistant. It also provides long-term immunity and extended periods without the detectable virus. Now, there are also a lot of challenges and considerations with this treatment. First of all, being finding suitable donors. There's a high criteria in donating and giving bone marrow stem cells. There are also a lot of potential complications, including graft versus host disease and graft failure. And the main problem with this is that there are different strains of HIV, like the CXCR4, which acts similarly to the CCR5, except the HIV will bind to the CXRC4 instead of the CCR5 gene, meaning that if this patient were to get the delta 32 mutation, it would not work the same on a patient as it would on a patient with the CCR5 strain. Now, there are also a lot of future directions and opportunities with curing HIV. Scientists are currently working on finding a globally accessible cure. It will cause a reduction in HIV transmission and an overall improved quality of life for these patients, as they will no longer need to be taking antiretroviral drugs daily and will no longer have to be afraid of HIV taking over their body. And here's a link to my essay.
We finished in about 5 minutes, so we have 5 minutes for any questions. I have one question. We can start off. Can you tell them what graft versus host disease is, or all that spiel that you said there, in case they don't know?
Graft versus host disease is when the tissues reject your body, and graft failure is when your body rejects the tissues after getting a bone marrow stem cell transplant.
Any other questions?
What is the difference between CXCR4
All the CCs.
No, it's just different genes. So HIV can bind to a different, like can bind to like this gene or this gene. It depends on which gene you have in your body. Yeah.
Which form of HIV is better, like less harmful, between the two that you mentioned?
So there isn't really an HIV that is less harmful. HIV just very quickly can spread through your body and develop into AIDS, which basically has a 100% mortality rate.
Okay, we have another question here.
If your body rejects the bone marrow, what happens to your body?
If your body rejects the bone marrow, you will need to get there's a long-term monitoring and recovery period where doctors will be constantly testing you and making sure that your body, doesn't reject what they gave you. That wasn't really related to my question, so I didn't go into much research on the rejection, but most likely the doctors will have to find a way to prevent your body from rejecting the bone marrow.
Any other questions?
We have back here, but here, let's go ahead. Same time. How can HIV be dangerous?
So HIV is a viral disease, meaning that it very, very quickly goes through your body and it makes you very sick. And it basically just will slowly, you just slowly start deteriorating until the HIV fully takes over your body and you can end up dying.
Which brand of HIV is more common? Like the first brand, like CC?
Of the C's that you were mentioning, which one do people have more genetically? So who has? Is that what you're trying to ask? Like who has? Okay. So of the CCs, what's the population look like? So like, do we all have that CCR5? Do we all have both of them? Do we have a combination? One together?
So it fully just depends, but most people do have the CCR5. But CCR5 and CXRC4, they're both related to the HIV1 strand, which is the most common.
How do you donate bone marrow?
You can go to like a local doctor, or you need to go to a donation place, and you have to be tested. You have to do a high screening process, and they will end up taking the bone marrow and giving it to whoever needs it.
If you get HIV, there's a 100% mortality rate. Like, how long do you actually live?
Well, currently, I said that in the 1980s there was a 100% mortality rate, but currently, if you're taking the antiretroviral drugs, the HIV will not spread through your body. But if you're not taking the antiretroviral drugs, it just fully depends on how strong your immune cells are and how long it takes for the HIV to fully spread.
Any other questions? We might have one or two after this.
Are there side effects of not if HIV can't bind you?
No, there's definitely.
So the 9%, he's talking about the 9% slide where they bind to you. He wants clarification on that. So go back to that slide, the 9%. Is that what you were asking about? Is there side effects of having that mutation?
No, there shouldn't be any side effects. The HIV just will bind to your body and will end up passing through.
So basically, you're immune to getting HIV. That's what you're saying. Okay. This will be the last question.
Okay, so concerning genes from genes, are they like genetically transferred? If my father has some gene, is it likely that I want to?
Yes, it can be genetically passed down, but it's not very likely.
Well, so this is not her research topic too, so she might not have any data on that. But genetics, they get passed down. It just depends on who gets it. Any other? Well, I guess that's it. We don't have any other questions. If there are any other questions at the very end and we have some time, we'll open it up to everyone. So Olivia, you want to introduce the next person?
I would like to introduce my friend and classmate, Simone.
Okay, so my name is Simone McGruel from class 716, and my focus question is, how can bone marrow stem cells affect the prognosis of individuals suffering from smoker's lungs? Bone marrow stem cells affect individuals suffering from smoker's lungs by reversing symptoms, replacing damaged lung tissue, and preventing lung cancer from occurring. So many of you might be asking what smoker's lungs is. So it's a collection of 4 main diseases, including COPD, emphysema, bronchitis, and lung cancer. So these 4 diseases affect over 20 million people every year in the US alone. And essentially what these diseases do is when you're exposed to high debris, dust, places like a construction site, or if you smoke or vape, your lungs begin to become inflamed, and your body reacts by lining your lungs with a mucus to help prevent these debris from coming in. But what this mucus does is it can make your breathing slower, harder, it can make you cough, make you be lightheaded, you might need assistance, and if your breathing gets bad enough, you will be put on an oxygen tank to allow you to breathe. breathe. So all of these diseases are very harmful for you, but if you continue to smoke or vape or be exposed to construction sites or high debris, it will become lung cancer, which is 32.7% fatal in men and 27.3% fatal in women. And either way, if you get lung cancer, you're going to go through a lot of chemo treatments, and it's going to be very painful and very discomforting throughout the rest of your life. So specific bone marrow stem cells I will be talking about are spongy tissue that they usually find in your leg bones, your arm bones, or your hip bones. And the specific mutation I will be talking about is the fibroblast mutation, which is a regenerative mutation in bone marrow, which helps with healing and any other problems that you may be facing in your body. So the process of the bone marrow transplant is extremely simple. First, doctors and scientists go into your lungs and they remove any damaged parts, any cells, and any just dangerous debris that are still roaming around in your lungs. Then they take these bone marrow stem cells that they have found in your bones and they inject them into your central bloodline. Now, these bone marrow stem cells have the ability to sense where problems are occurring in your body. So they navigate themselves to problems of area, specifically in your lungs. And once arrived there, they can face any diseases and replace them with helpful cells to get rid of the harmful ones. And throughout time, these bone marrow stem cells will just stay there, create extra shields of help, and just prevent you from getting more and more sick from these diseases. So the bone marrow transplant has been around since the early 1930s, but the bone marrow transplant for smokers' lungs was only recently discovered in the late 2010s, and it was produced when a professor named Wei Zhu in China gathered 20 patients with a variety of mild smokers' lungs, extreme smokers' lungs, and all different types of smokers' lungs, and tested this bone marrow transplant on them. 85% of them received the fibroblast mutation that I mentioned earlier, and either way, all of their conditions extremely improved, and they were able to breathe on their own. Some of them were taking off their oxygen chains, and they were just overall living a better, happier life. So after this news was spread worldwide, a professor named Jaden.
Pardon the interruption, please. Can Ms. Esposito dial 2093? Ms. Esposito 2093. Thank you.
Don't mind that. Anyway, so once this news spread to the U.S., a professor named Jaden created the two principles of this bone marrow transplant to see and test if this bone marrow transplant would succeed or fail. So the first principle was the cleaning of the lungs, because what would happen if they then left these harmful cells and dangerous debris, they could possibly transfer these harmful diseases to the new cells, which might inflict the problem and make it worse. The second principle was the bone marrow stem cells being able to navigate themselves to the correct area. If not, then they might not heal that area and they might need to redo the transplant, or it might not be possible for them to do the transplant again. So lung tissue is the tissue that makes up your lungs. It also helps with containing the alveoli, which allows you to breathe and filter oxygen through. So through tests, doctors can see how your bad or worse condition your lung tissue is in, and you can see it by a darkening color, or if it's just not breathing properly. So once these bone marrow stem cells are sent in, they have a regenerative mutation that can help reduce inflammation of the lung tissue so that your bronchial tubes are able to pass through more oxygen, and they can also heal with any wounds or scars that have been created in your lungs from smoking, vaping, or the debris you have been exposed to. So obviously I mentioned symptoms earlier of not being able to breathe, doing things on your own, maybe just not being able to do things that you normally do, jogging or playing sports or anything like that. So what these bone marrow stem cells are also able to do is breathing is what helps us bring in oxygen and stuff like that. So without our oxygen being filtered through and coming in as it normally would, we're not going to be able to do stuff as we normally would. We're going to need assistance. But these bone marrow stem cells are able to make it so that we are getting the correct amount of oxygen, which should be. And because of this, you're not going to feel lightheaded, you won't be able to breathe normally, you're not going to need assistance in most things that you have been doing. Okay, so obviously I mentioned before that these diseases can become lung cancer if you continue to follow the process of vaping, being exposed to smoke, or anything like that. So through MRI scans, doctors can tell if there are any lung cancer cells that have been created on your lungs. Now, these bone marrow stem cells are not able to get rid of the lung cancer cells, but they are able to prevent them from spreading. So these lung cancer cells multiply and can take over your lungs, but the bone marrow stem cells are able to contain these lung cancer cells in one area, so they don't spread and make your condition worse. So complications. Obviously, if you continue to smoke or vape or be exposed to these debris or construction sites, you're not going to improve. You're going to get worse, and you might have to get the transplant done again. Also, in the test, rashes were formed on the patient's necks for a couple of weeks, but they usually died down. It was just a reaction to the heat that was being exposed to them. Additionally, if they are not able to find correct donors with the fibroblast mutation, they might not be able to give to this person, and their condition might worsen, and it might be too late for these bone marrow stem cells to come up and help them. Okay. So, additionally, in these tests, 53% of patients with mild smoker's lungs, so around emphysema or bronchitis levels, were fully cleared of the disease in testing. 35% of people with extreme smoker's lungs, so right on the cusp of becoming lung cancer, were fully cleared of the disease. And 76% of people with both mild and extreme smoker's lungs were fully cleared of their symptoms like coughing, vomiting, not being able to breathe well, and other stuff like that. So finally, the worldwide impact. All of these tests that have been done have been beginning in China, but they are slowly moving to parts of Europe, Asia, and Africa. North and South America have the most restriction on stem cell usage laws, so it might take a while for them to receive the testing and the permits to do these things, but it is slowly helping people all across other parts of the world, which is very good. So the bone marrow is able to remove some of the lung cancer cells, but they're obviously not going to be able to remove all of them if they've spread thoroughly. But if there's not too much, they are able to contain it in one area where if you are getting surgery or some sort of process, they might be able to salvage your lungs and replace it with a donor's lungs. What they do is they do surgery. So what they usually do is they'll cut out a piece of your skin, but then they'll take this special tool that will inject itself into your bone and remove the stem cells that make up your bone.
They do essentially break a little piece of your bone to remove the bone marrow and then transfer it to the patient who's receiving. One more question?
After you've gotten the bone marrow transplant, does that affect your addiction?
It may not. Obviously, smoking, vaping are a very addictive thing. But I feel like after that, the person has been like, oh my god, this is not helping me, they might look at themselves and be like, oh, maybe I should stop this, especially because it would reverse the health of the bone marrow.
Great job, Simone. Can we introduce the next person, please?
Okay, yes, I would like to introduce my classmate Jaden.
My name is Jaden Luke, a
class 716, and my focus question is, how can stem cell therapy affect tissues? My thesis statement is, stem cell therapy has a beneficial effect on tissues by reducing inflammation during the healing, replacing the injured parts of tissues with new ones made of stem cells, and harvesting stem cells that decrease the risk of re-injury. Stem cell therapy is a rehabilitation that regenerates injured tissues using stem cells. And I know you're wondering, what are stem cells? Well, stem cells are undifferentiated cells of multicellular organisms. Undifferentiated means that these stem cells have the potential to become any cell in the body, and these cells could then become into any tissue. And the photo applied here just shows how different units can form into different complexes. How it shows how cells can become tissues, tissues can form into organs, and organs can form into organ systems. This will then turn into the whole body. The important ones that I'm going to show is how cells can become into tissues. Stem cell therapy can be beneficial in many ways. One way it can benefit the body is by reducing inflammation. Scientists use mesenchymal stem cells as part of the process. Mesenchymal stem cells are typically found in bone marrow, and the function of mesenchymal stem cells are to release cytokines. And cytokines are just proteins that make connections and signals with other systems. Additionally, once they are activated by mesenchymal stem cells, that their responsibility is to signal the immune system, and the immune system will then decrease inflammation. There is a special cytokine that's activated, and it's called interleukin-4, or IL-4. Interleukin-4 induces white blood cells to initiate attacks on inflammatory causes. And there's a special white blood cell that's activated, and it's called neutrophils. And neutrophils is a white blood cell that is the first line of defense for the immune system. In order to replace injured tissues, scientists need something to return it to its place. Scientists conserve pluripotent stem cells, which are the stem cells that can potentially become any cell in the body. And pluripotent stem cells will be injected into petri dishes. And petri dishes are just plastic or glass dishes that have covers in it. And this is a picture of a petri dish. It shows how there's like a cover, and the scientists injecting the pluripotent stem cells inside the petri dishes. And when the pluripotent stem cells are inside the petri dishes, they'll undergo blastocyst complementation. This will get the pluripotent stem cells to grow into a specific tissue that it needs to replace. In the petri dish, this pluripotent stem cells will grow and divide till it forms one complex unit. And this typically takes around 2 to 5 weeks. The pluripotent stem cells are modified to behave just like somatic stem cells. So since somatic stem cells are in many of the tissues and all the major organs, the replacement process will be easier. And the photo I've shown here just shows how the locations of somatic stem cells in the body. It shows how it can be in some of the major organs and most of the tissues in the body. And once the pluripotent stem cells are finally finished going through asymmetric division, they are going to be injected into the body as new cells. And asymmetric division is just the process of the stem cells growing and dividing in the petri dish. Before injecting the induced pluripotent cells to the body, scientists have to apply dextrose. And dextrose is just a type of sugar that can be a huge source of energy. Dextrose is a type of sugar that can be a source of energy. It's crucial because losing cells in the body could deplete the energy of the body drastically, and stem cells in the body could be used to function energy. And the photo I've shown here just shows different levels of blood glucose levels or energy. One I've shown hypoglycemia, the low blood sugar, the normal level, and hyperglycemia. Usually when people apply dextrose to the body, it could be an overflow for the body. The body could be functioning energy too rapidly. But in this case, when you're losing energy really quickly, you need the body to be in hyperglycemia with dextrose. Then the scientists just inject the somatic stem cells out of the body so that they can inject the new stem cells, which is the induced pluripotent stem cells, back into the body. And according to the National Institutes of Health, stem cell therapy decreases recovery time by about 30%, which is a significant amount because recovery time could be a long time and 30% could be assistance. With scientists that perform stem cell therapy has had a 79% success rate. And this photo just shows the stem cell market and how they're predicting in 2032, the stem cell therapy market will be $43 billion. As obtaining new tissue can increase the strength of that specific organ that it replaces. And as muscles age, the muscles can decrease in youth and the ability to contract in size. The ability to contract in size just means the ability to grow in size, get wider or bigger. And the photos I've shown here, it just shows, before scientists use these therapies and these surgeries on humans, they have to set trials on animals first. So they perform stem cell therapy in mice before they use it on humans. And it shows how the regular, which is the young muscle, the typical amount of muscle, the age muscle, and the rejuvenated muscle. The black dots are supposed to represent the body's system in holding the muscle. The young muscle has the typical amount. The age muscle seems to have decreased in muscle. And the system seems to not uplifting itself. But the rejuvenated muscles or the regenerated muscles, which is from stem cell therapy, the new stem cells, they form tissues, and these tissues seem to have the best system in holding the muscle, and the black tops seem to be increasing in size. Therefore, the new regenerated muscles is sturdier, so they'll be more difficult to injure or bend or break. And this photo just shows the important parts of tissue. It shows the tendon, which is sort of the outside part, the ligament inside the tendon, and it shows the joint cartilage in between the bones to form the structure of the tissue. And these are my sources.
Questions you might have? No questions? Any hands up? Any questions?
Yeah. Yeah. Interleukin-4 could be used for chemotherapy, but I've shown it here for reducing inflammation. This is the function that I've shown what interleukin-4 does. It's a type of cytokine too.
Any other questions? Oh, I see a hand up. Do I see a hand up? No, we're just stretching. All right, I guess we can move on to our next presenter. Would you like to present them, please?
Yeah. I'd like to introduce my peer and friend, Samuel Tarlow.
How can stem cell treatments affect people with head or neck injuries? Stem cell treatments can affect people with head or neck injuries by improving impaired vision, improving brain function, and regenerating neurons. First off, what are stem cell treatments? It's just when you treat an injury or condition with stem cells. And mostly today I'm going to talk about mesenchymal stem cells, which Jaden started to talk about. And mesenchymal stem cells can replace damaged cells, urge the immune system to replace damaged cells, as well as urge progenitor cells to develop into fully developed or fully mature cells. So as you can see, a stem cell can become another stem cell or any type of specialized cell in the body, but a progenitor cell can only go into one specific group of cells. So it can become any type of neuron, but only one neuron. So what injuries am I going to be talking about? Chronic traumatic encephalopathy, or CTE, is the main symptom is brain degeneration. As you can see, the brain loses a lot of mass between the normal brain and advanced CTE. And traumatic brain injuries, or TBIs, which is just the terminology for a substantial injury to the central nervous system, the central nervous system being the brain, eyes, and neck. So this research is important because the brain is such a fragile and complex organ. It's very difficult to repair itself, and it's very difficult for us as humans to repair it. And as you probably know already, most treatment options have been gravitating towards stem cells, but neural stem cells, which is what would be found in the brain, are very rare, hard to find, and in the nooks and crannies of the brain. So this was a study run by the University of Tennessee in which the eyes of mice were indirectly damaged in the same way that a traumatic brain injury would. And then these same eyes were injected with a mesenchymal stem cell serum called ASC-CCM into the vitreous chamber or intravitreally. And then they used Muller cells to track damages. Muller cells are a type of glial cells. And what glial cells do is that they maintain the environment of the neurons. So if there's more Muller cells in an area, you can tell there's more damage. Directly after injury, the mice eyes had a higher level of Muller cells than average. But then after treatment, in about 2 to 3 weeks, the level of Muller cells got down to average. And it said in the study that you couldn't see a difference between the eyes of the mice that were injured and then treated and the eyes of the mice that were just like nothing happened to them. So neuroinflammation can decrease neuron function, which if it's in the eyes could then decrease vision. And B-wave amplitudes is the distance between the highest and lowest point on the chart of pupil dilation. So this very crude diagram shows a B-wave amplitude. This black circle, if that's the smallest that your pupil can get, and if the gray circle is the largest your pupil can get, then that white line would be your B-wave amplitude. A higher B-wave amplitude would be better because it means that your pupils are better at reacting to drastic changes in light. So a simple eye test is a great example of a test for visual acuity as well as contrast sensitivity, because it allows you to test for the ability to pick out small details and the ability to see the sharp outlines of the letters. So after the mice were injured, all of the symptoms that I just showed you decreased drastically, so there was a lot more neuroinflammation, lowered B-wave amplitudes, lowered visual acuity, and lowest contrast sensitivity. But then after they were treated with this ASC-CCM, those symptoms were reversed, and as I said earlier, it was nearly impossible to tell the difference between the eyes of mice that were injured and then treated and the eyes of mice that weren't injured at all. So this was a study run by Jocelyn Bloch, a neurosurgeon, and Jean-Francois Brunet, a biologist, in which the brains of monkeys were injured or lesioned. A piece of the brain was removed and then cultured by putting the neurons in a petri dish, and then they would reimplant the cells that developed. So you can see in these pictures, the cells that developed from the culture looked nearly identical to a neural stem cell culture. You can see there are larger, more mature cells being surrounded by smaller, immature cells. So after the monkeys' brains were injured, or before they were injured, they were taught to do a simple dexterity task where they would remove food from a tray. After injury, their performance went down to 40% of their previous capacity, and after treatment, that performance went back up to 90 to 100% of their previous performance. Separately, as I said earlier, CTE degenerates the brain, and so if we have the ability to regenerate these neurons that are lost, it could reverse the symptoms of advanced CTE. So this study was done by BioAccelerator, which is a medical clinic, in which they treated a TBI patient with mesenchymal stem cells. Their motor control improved as they were able to start climbing as well as descending stairs with little to no assistance. And their speech improved. They were able to start speaking slower, more deliberately, and self-correct when they made a mistake. Separately, Boulder Biologics, which is a medical clinic in Boulder, Colorado, treats a lot of patients with traumatic brain injuries as well as CTE, and in like half the cases, or 50% of the cases, speech, memory, cognition, and motor control improved, and there was a very, very low chance of adverse side effects. That's it.
Excellent job, Jaden. Okay, so if anyone has any questions for him, he has the floor for 4 more minutes.
Let me just repeat it. How bad is CTE and TBI?
So TBI is not like one specific injury, so I can't really give you much information on that because it's just traumatic brain injury. It's an umbrella category. But then CTE is very, very harmful, as you can see. I mean, this is highly advanced CTE, so it's been going on for a long time. But all of those neurons lost had some function originally. So like if a section of your brain that's responsible for memory got degenerated, you wouldn't be able to remember things. And it's also, if enough of your brain gets degenerated, it's fatal.
There's another question over here.
If you have CTE or TBI, what's some side effects or like symptoms?
So, as I just said, like if the section of your brain that's responsible for memory gets injured, then you could have a decrease in memory. Or like if the section of your brain responsible for motor control gets injured, then you might have a harder time moving. You might even need to be put in a wheelchair. And traumatic brain injuries, same thing. It could injure sections of the brain that are responsible for important things, and all of these things could be fatal if the right section of the brain gets injured or degenerated.
Any other questions?
Yes, in fact, I originally wanted to do this topic about football players, but I just couldn't find enough connecting evidence, so I had to move away from that. So it's less of a disease, and it's not like it's not like something that's attacking your brain. It's just that it gets hit. In this case, it gets hit so many times that the neurons just like get damaged and then eventually get destroyed. Anything else?
Okay. All right. Well,
Now we have a panel of people who are going to answer some questions that you guys submitted. So please welcome Smith Matthews, Melody Lee, Billy Quach, and Paris Simmons.
Hi, I'm Smith Matthews. My focus question is, how is lab grown meat superior to conventional meat? But today, I'll be answering the question, how much time do you spend making the presentation submitted by Ingrid Matthews 601? I'd say that making the presentation, depending on how motivated you are to do it, can take around 1 to 2 weeks. However, getting there can take a while, and you'll need about 5 months to prepare note facts, research your topic, and make sure that you're doing everything nicely and well organized. Only then, after you're done with that, you can start your presentation. Okay. Now I'd like to welcome my colleague and classmate, Melody Lee.
Hi, I'm Melody, and my focus question was how can bringing back the passenger pigeons affect its local ecosystem? But today I'll be answering some questions from how was your experience with double IAM and what was the most difficult part of it? From Claire Chen, class 603. IIM has been an incredible experience for me, but it has also been difficult at times. IIM has provided me with numerous benefits and enabled me to acquire additional knowledge and skills. It covers abilities in data analysis, leadership, communication, and problem solving. IIM gives me a head start in supporting my career success in the future with the power of using formal writing, presenting, and more. However, it gives a few challenges, such as when I was writing my formal essay. It took me a long time to consider the ideal terms to use or even how to come across as a professional when I was writing my essay. Presenting to the class was part of that as well. I was nervous, stuttering, and anxious. It was really painful. It was really painful to speak in front of the class. Yet the experience had helped me develop my public speaking confidence and communication abilities. And the next question is, how do I present in the smoothest, clearest, best way possible? From Yi Shin Lin, class 604. To present your slides in the best and smoothest way possible, you have to start off by presenting your slides to your family or friends to gain some confidence and understanding more of your topic. Have your slides visible and clear. Have index cards with you in case you forget what you're going to say. Always remember to engage with the audience and have a loud voice and good pace. Done. And now I'm going to welcome my classmate, Billy Clash.
Hello. Hello, everybody. My name is Billy Quatch, and my focus question was, how can advances in the powerlifting field affect the organ transplant industry? But today, I will be answering 3 frequently asked questions. My first question is, do you think 7th grade IIM is difficult? From Alan Chen of 601. Even though it may be challenging at times, your teacher will be available to support you throughout the way. It will be simple, first, choosing a topic, looking for sources, and so on for the first few months. It will gradually begin to get more difficult, with the need to write an essay and finish your presentation and more. My second question is, what helps you present in front of people? From Fiona Lynn of 602. Some helpful suggestions for presenting in front of people. Presenting in front of a large group of people are to practice, maintain eye contact, and speak clearly with an appropriate tempo. Though it may be challenging, try not to read directly from your slides or index cards, and rather use them as a crutch when you need them. A few tips to help you if you begin to feel anxious during your presentation are to take deep breaths, concentrate on your material, and instead of letting the fear of failure worry you, convert it into enthusiasm and make the best impression. My third question is, what are the expectations from Miguel Acosta of 604? There are many expectations and high standards in the 7th grade IIM class. There are tight deadlines and a lot of work that is required to be completed, which could begin to become stressful. Within your allotted time, you must select a topic. Complete your research, compile your notes, and finish your essay and presentation. With that, I would like to introduce the final person answering questions, Paris Simmons.
Hi, I'm Paris Simmons. I'm the final panel member. I made the slides, and my focus question was, how can act laws affect human tissue generation research? First question, what websites do you use to get your information? Submitted by Samantha Zing in 601. In 7th grade IIM, we utilize a variety of resources, such as magazines, journals, newspapers, which can all be found on the McKinley Formatting database. We also use reliable websites and YouTube videos, but those cannot be found on the database. Second question, what was the hardest part to do in your presentation? Submitted by Litzy Moreno in 603. In the STEM tools unit, we have to do a 5-paragraph essay, and it gets a bit hard when you have to put all the information you've gathered into about 5 or 7 slides. and eventually you'd have to talk about the topic, which is also hard. And then, final question. What are the pros and cons of 7th grade IIM? Submitted by Jaden and 601. So the pros would probably be that the program looks good on the high school applications, you learn how to write formally, and you learn about interesting science topics, such as stem cells and science. So the cons would probably be it's hard to keep up the work, you have to manage, need a good time management schedule, and you're expected to have high standards than other classes.
Great job, everyone. All right, so I'm very impressed. I hope you are very impressed. We have about 15 minutes remaining, and now it's open for Q&A. Whatever questions you have, so we have our 5 presenters. You can ask them any questions specifically about what they presented, or you can ask the panel members about what you're interested in 6th grade, going into 7th grade, what your fears might be, what you're expected to do. So it's an open Q&A, whoever has any questions. But first, Cummings?
Well, I don't have any questions, but thank you so much, everybody. I'm super impressed, and I know everybody else is. So let's give all of our presenters a round of applause. And we know that the 6th grade IIM classes submitted over 100 questions. And what did they answer? How many? A dozen? So I know that some of your questions didn't get answered. So now is your chance. As Mrs. Leonardo said, we have about 15 minutes for you to ask questions. Mr. Gibson, so Mrs. Leonardo, just let Mr. Gibson come on up. He's going to help us bring out some chairs. And we're going to put some of our IIM presenters up on the stage so it's easier for you to feel like you can ask questions. So right now, I want you to think, what did you ask last week that did not get answered? Okay? This is the winning slide. Winning. All right. Yes, there are bright lights on the stage. So we have our presenters up on the stage. Remind me, so go down and remind us of your names. Let me pass this mic over to you. And then we're going to get...
I also have the Q&A panel up here. If you guys have any questions, they'll stand up and they'll answer for you too.
And there's always Mrs. Leonardo's too, if you want to ask her questions, 6th graders. And all right, so let's get some questions going. Who's on the question they want to ask? Can you stand up and say it very loudly?
Was it fun doing it? It was very fun. It was definitely an enjoyable experience. At certain times it can be stressful, but overall the finished product is very fun.
Does anybody else want to add?
Olivia, how did you pick that subject?
HIV was interesting to me, and I was very interested in HIV and stem cells. That's why I picked the topic.
I have a question for you over here. Stand up and say it very loud. I can say it for you. How do you get HIV?
So it is blood. It can be given through blood. It can also be vertically transmitted and through the mucous membrane. So you can get it from most commonly sharing needles, and obviously it's vertically transmitted too. And it can be passed down from mother to child.
Are there any side effects on taking HIV medicine?
HIV medicine side effects?
There are no side effects to taking antiretroviral drugs.
Go ahead. Can you repeat the question when you went?
Can you repeat the question? Throughout the process of IIM, we will have to research the topic. I don't know if there was any key things that we would have to know. What do you mean by that? About our topic or just about the? Oh, I wish I knew more about these new treatments because it's really interesting to research about it, about the side effects and the effects.
Anyone else on the panel want to add on to that?
So one thing that I kind of wish I knew is just what to do when you're standing up in front of people, because I'm sure you noticed all of us were kind of fidgeting, and if we had something to do while we're standing up in front of you guys, it probably would have made the whole process a lot easier.
And I have Melanie wants to add on to that. Do you want to add on?
Besides that, we also have the essay. Besides presenting to the class, and writing essay was also a key thing that you have to always remember how to sound like a professional and use complex words, and always remember to have 5 paragraphs, and remember to keep your essays in order and sound more clear and visible.
Any other questions?
Yeah, we'll get around. How do you make the graphs or the data table images?
Me personally, I didn't make the graphs. I basically went on Google and typed specifically what I needed, and I would look through every site to see if they had a graph and all the information needed.
Do you know anything about forensic sciences?
We haven't started on that unit yet, so no, not really.
We're starting later this month.
All of you guys are so well spoken. I admire all of you. But just give me a little tip. How do you guys stay so assured and so responsible and professional under pressure?
Honestly, no clue, but I guess we practiced a lot in front of the class, and we gave each other, our classmates gave us tips to keep concentrated and improve our presentations throughout time. Thank you, Judy, for that.
Can you regenerate parts of your brain from traumatic brain injuries using those stem cells?
Could you repeat that?
Can you regenerate parts of your brain from traumatic injury using stem cells?
So yes, I said in my presentation, they were able to create almost pseudo stem cells by taking a piece of brain, and then I forgot to mention this, but when they reimplanted those stem cells into the damaged brains, they ended up staying in the brain and even developed back into fully matured neurons.
What was the hardest part of your research?
I think the hardest part of the research was was mostly in the beginning, with just finding sources that didn't give me all the same information and were very repetitive, and also having to gather all the information, put it into note facts and subtopics, and then putting it onto our slides.
Another question? I have a question over here. Stand up and say it loudly.
Could you use genetic modifiers to make the mutations in the bone marrow?
That wasn't in my topic, but it could be possible.
We have another question over here.
Would you do it again? The whole not unit, but would you do a presentation like this again if you had the choice? again because presenting is I like informing other people, the researching. The whole process was really informing. It was really entertaining, and I would love to present again.
I would say also yes, if I were given the option, I would do this again because it's really interesting to be able to find your own topic and then go and research it on your own rather than being conformed to the school's curriculum. So I would say yes, I would do it again if I were given the option.
Stand up.
If you lost a part of your brain and then the stem cells regenerate it, if you lost memory, would you have the memory back again when you regenerate it?
So I didn't go far into after the treatments other than just basic level results because this treatment is very new and so there's not a lot of data on it. But I would assume that no, your memories would not be restored. I think you would be able to store new memory. If, for example, the symptom was short-term memory loss, I think that your short-term memory would improve and you would be able to start, you would do better with remembering things earlier, but I also don't think that the memories get erased when the neurons degenerate, but I'm not entirely sure, so don't take my word for it.
But that's a good question, and that could be a potential focus question for next year. So this is a good opportunity to listen to what they have to say and anything that's lacking in their presentation, not because it wasn't good, but because that wasn't their research topic. That could potentially be something that you might be wanting to research next year. So that's a good question. I saw another hand up.
So let's say you do get treated with stem cells and they restore part of your brain. Would that part of the brain that was restored be as effective as the last part of the brain before it got damaged?
Once again, I didn't go too far into that, but I would say yes, it would be about the same. It would be about the same performance. In the monkey test, it was dexterity that was injured, and once the neurons were regenerated, their performance went back up to 100% of their previous performance.
After mastering this topic, do you feel like you would use it in further careers?
You said after doing this, would you use it in other careers? Yeah, definitely. You mean for the speaking or the actual presentation, the information that I researched? All the what? All the skills. Oh, yeah, definitely. Because in future jobs, let's say you're giving a meeting or something, you definitely do need public speaking. You can't just be stuttering the entire time and not knowing what to do. You need to have confidence and you need to know your information.
There's another question over here.
With every topic that you guys research, is that involved in the career you want to have when you're older?
So I can't speak for everyone here, but me, when I started on my topic, I was doing it to look into athletes with head injuries because my dream is to be an athlete, but I wasn't able to go into that. There wasn't enough evidence. So for me, yes, it did have something to do with a further career. I can't speak for anyone else.
I was also trying to research how my stem cell therapy can affect athletes because I want to be an athlete when I grow up, and I was just trying to see if these therapies and these surgeries and these treatments could treat athletes and just decrease the recovery time. But I didn't do it specifically on athletes. I did it specifically on just anyone. So I did.
Well, me personally, when I grow up, I want to be a chemist and I want to research medicine. So CRISPR and these other genetic editing tools are definitely helpful because they can help cure diseases and make a lot of the patients healthy.
Okay,
like these little scare guys.
For me personally, it was just based off family members that had the disease. It really didn't go on into anything of a career for mine.
I did want to be a doctor, Olivia. Thank you for pointing it out. For me, HIV, what I researched isn't really related to what I want to be what career I want to have, but I was just very interested in my topic.
Okay, we have someone else that wants to add on. So she's going to tell you her focus question, what she was interested in.
My focus question was, how can the cosmetic use of rose stem cells affect the appearance of facial tissues? And when I grow up, I do want to be like a cosmetics doctor to like help with skin or like a dermatologist. So it kind of did affect my career.
So, but you don't necessarily have to choose something that has to do directly with what you want to be when you grow up. But it's nice. It's an opportunity to check it out. Any other questions out there?
I'd just like to add on to that last comment, and you are all young people exploring different things. All of the skills that you learn in IIM, from research to writing to presenting, are going to be useful no matter what you choose to do for a career. So you don't have to put that kind of pressure on yourself to think about, career-wise. It's more what catches your interest right now, and you go a little bit deeper into it, and then you see where that takes you, okay?
I agree. Any other questions from anybody in the audience? There was a lot of questions. We got up 100 of them.
What part do we enjoy doing the most? Honestly, for me personally, my favorite part was making the slideshow because I enjoyed being able to kind of have a little bit of creative freedom with what I was able to make and being able to put my information on the slides and see how far I've gone from the beginning to now.
So I personally enjoyed the part of choosing our topic, of researching all these different type of variety of different topics about stem cells, because I like informing myself about different topics of stem cells, and I thought it was fun trying to get your own topic and what to research.
Weirdly enough, my favorite thing about this was actually writing the essay. Me personally, I have a bunch of ideas in my head constantly, and it just felt so good to just put all of them down. I just wrote everything that was on my mind, and it was it was amazing. And I didn't have to dumb it down for Miss Leonardo's because she's a very intelligent teacher.
Definitely not for the grade, that last comment. My favorite part was researching my topic after I'd chosen it because I personally really enjoy informing myself on a particular topic, that especially when I was able to choose it myself in any research project scenario. So my favorite part was going into it, finding sources, finding no facts and stuff like that. Simone, want to add anything?
I have to agree with Olivia, the presentation. It's really nice trying to find all the things, make it look good. It definitely takes a lot of work because you want to make sure it's accurate information, you want to make sure it's not too much, you want to make sure that it's attractive, and you want to make sure it's a good presentation. But that was my favorite part.
Okay, I think we're going to wrap it up. I just want to say thank you to everyone. These were great presentations, but I had a lot more to choose from in the class. They did amazing in the classroom, and you guys will do amazing in the future. So I just want to give a round of applause to everyone.
You're welcome.
Awesome. You're the best. Have a good day. Have a good day. Thank you. gonna have to do this too?
No, we're gonna do something different. We'll talk about it. All right, I'll see you all. Folks, if you didn't check your grades 8-16, check your grades. See me at lunch if you want to see me at lunch. I believe so. It's giving me an error.
Yeah, I saw that before.
I don't know.
Maybe we should close out.
Yeah, I was thinking.
Close
Yeah.
out just YouTube.